Hello everyone, welcome to Lecture 3. Again, I'm Jeannie Lee. I'm a professor of genetics at Harvard Medical School. I'm also a faculty member in the Department of Molecular Biology at the Massachusetts General Hospital. All right, so we talked about the different steps of X chromosome inactivation. In Lecture 2, we covered in some detail the molecular mechanisms behind X chromosome counting and allelic choice. And what I'd like to do in this last lecture is to talk about the step of nucleation followed by the spreading of silencing across the rest of the X chromosome. And recall that the problem is that here we have Xist RNA, which is shown in red, and it coats the inactive X chromosome in cis, and it does so without spilling over into any of the other X chromosomes in the cell, or to any autosomes for that matter. And so the question is, how does this RNA stay put on that chromosome? And we know that this property belongs to the small piece of DNA called the X inactivation center, because when we place the inactivation center on an autosome, so a non-sex chromosome, we see that Xist can also coat that chromosome in cis without spilling over into any of the other chromosomes, including the X chromosome, where it normally would be produced and would be spread. Okay, so we're going to start with this concept of nucleation. Now, this is something that we stumbled upon sort of by accident. So recall this experiment, which we had done back in the 1990s, that sort of led to this dogma that Xist RNA is strictly cis-acting. So the idea here was to place an inactivation center, or Xist, onto an autosome in a male cell, and we could observe that Xist is produced from this ectopic inactivation center, and that RNA spreads strictly in cis across the autosome without ever diffusing, or so we thought, to the natural X chromosome, which is located in a different place in the genome or in the nucleus. Okay, so that has been recapitulated many times by various studies. But then many years later, when we repeated the same experiment, except this time we did it in female cells, and in the post-inactivation state, we got a very surprising finding. So now here are two X chromosomes, one's inactive and being coated by Xist RNA. We place an inactivation center, or Xist, on an autosome, and we saw, as we expected, that Xist would be upregulated and would coat and silence that chromosome in cis. However, what we didn't anticipate was that within 24 to 48 hours of doing this, Xist RNA started to fade away from the inactive X. Now, upon probing deeper, we realized that Xist wasn't actually going away or disappearing. In fact, what was happening to it was that it was being pulled away onto the autosome, which had this multimerized X inactivation center. So that was extremely puzzling. And here's the actual experiment where you can see the autosome very nicely producing Xist, and the X chromosome down here, which is starting to fade away, at least with respect to Xist RNA. And within 24 hours, we see the transgenic Xist spot, but the X chromosome spot was nowhere to be found. So that was indeed very surprising. And what we learned from these experiments is that, in fact, contrary to what we were expecting, Xist RNA can diffuse in the nucleus. But we normally see it localizing in cis because there isn't another site in the nucleus that would be receptive to Xist. Okay? So Xist can diffuse in trans and act on a different chromosome. So this led to a revelation that there has to be a sort of a nucleation center within the Xist gene itself, and that these autosomal transgenes must somehow contain such a nucleation site. And so we went ahead and dug around to try to pinpoint this nucleation site and identified 3 binding sites for a transcription factor called YY1 that was immensely important to this process of nucleation. And so what we showed is that YY1 protein binds to these 3 sites and serves as a tether for Xist RNA to anchor to the inactive X chromosome. And when we mutated these 3 YY1 binding sites, look what happens to Xist. It disperses across almost the entire nucleus. So the RNA detaches from the X chromosome and floats away into the nucleus. Okay, so we could recapitulate that finding by depleting the cells of this critical anchor, the YY1 protein. You see how it recapitulates the loss of this Xist focus within these female cells. And it wasn't because Xist was no longer being produced. Xist was still produced, but it could no longer attach to this nucleation center. Okay, so we conclude that YY1 is a tether for Xist RNA at the nucleation site. So this nucleation allows Xist to attach prior to the process of spreading. It's an absolutely critical initial step to X chromosome spreading. So what we observed is that the nucleation site is located within or very close to this repeat sequence we call repeat F near the 5' end of Xist, that it contains a trio of YY1 binding sites. The YY1 protein is very important in tethering Xist RNA to this nucleation center. And so what we imagine is that Xist RNA gets produced from this genetic locus, and it co-transcriptionally loads onto the nucleation site. And what we mean by co-transcriptional is as it's getting synthesized, when this piece of the RNA gets exposed, it detaches or attaches to the inactivation center before the transcription is complete. Okay. And then from the nucleation site, Xist spreads in 3 dimension across the rest of the X chromosome. All right, so now let's talk about this concept of spreading itself. How does that actually happen? All right, so what happens from here, which is the synthesis of Xist and attachment to the nucleation site, all the way down to here, which is a chromosome condensation and a lockdown of gene expression on that chromosome, remains largely unknown. However, we do know that several things have to happen in between. One of which is that Xist has to push away these factors that normally give rise to gene expression, so the activating factors, and at the same time, it has to recruit repressive factors to the X chromosome to start the process of silencing. And then probably at the same time, it is generating a new kind of topology on the X chromosome. All right, so how does all of this work? Well, we know that Xist is multifunctional, and to identify important domains that are essential for spreading, we have performed a systematic deletional analysis of Xist. So here again is Xist, and what we've done is use the CRISPR-Cas9 gene editing technology to remove sequentially 1 to 2 kilobase regions from Xist RNA, and we do this at the endogenous Xist locus in female cells so that we can observe everything happening in the normal physiological state. And what we found are 2 repeats that are very important to this process of spreading. Okay, so there's repeat B, shown right there, and repeat E, which is in the last exon of Xist. So normally, as you know by now, Xist forms this very tight focus over the inactive X chromosome. But when we deleted either repeat B or repeat E, what you see is a dispersal of the Xist RNA cloud sort of across the nucleus. Okay, so I think that's very nicely illustrated here and here relative to the wild type Xist RNA. So to get a better understanding of what's happening at the molecular level, we performed an epigenetic method called CHART-seq, and this allows us to map RNA binding sites on chromatin. In this case, we're interested in Xist RNA. So here I'm showing you a time course analysis of Xist binding during X inactivation. And in this top row here, you can see that Xist is initially expressed from the X inactivation center, and it's nucleating there. But then it's spread sort of all at once to the rest of the X chromosome. In other words, it's not spreading sort of locus by locus in 2 dimensions down the chromosome, but instead it's spreading all at once in 3 dimensions, because you're seeing that the RNA is piling up across the X chromosome more or less at the same time. And the other thing we found out from this experiment is that Xist is first going to gene-rich regions, and in particular, it's going to active genes, exactly the genes that it ought to be attacking first, if we're talking about silencing of an entire chromosome. And then eventually Xist spreads over the entire X chromosome until it essentially covers the entire 166 megabase chromosome. Okay, so we believe that Xist RNA spreads in 3 dimensions across the inactive X chromosome. It first nucleates here at the X inactivation center, and then the RNA is transferred through proximity to various secondary sites, of which there are probably about 100 or so, first targeting the gene-rich, the active gene regions, before spreading to the rest of the X chromosome. All right, so what happens when we delete this critical repeat B, which is important for spreading? So again, here in the top row, we can see the wild-type spreading pattern that's covering essentially the entire chromosome. But when we delete repeat B, you see that there is a diminution of binding across the entire X. And it appears as though the ends of the X chromosome are being more affected than the middle region, consistent with this idea of a spreading defect in 3 dimensions. Okay, and as you would expect of an RNA that can no longer spread efficiently on the X chromosome, gene silencing is severely compromised. So here, by RNA sequencing, you can see lots of activity still across these 2 representative genes, and the activity shown here is these little red tick marks. And there's almost as much activity on the inactive X chromosome as on the active X chromosome. Okay. So now, we've also come to understand, in spite of what I just showed you by chart sequencing, that Xist covers the whole chromosome, when we looked at individual cells, okay, by super resolution imaging with a resolution of 20 nanometers, we see that Xist isn't actually plastered on the entire chromosome. It's not really a coat. It's actually a cluster of about 100 dots, with each dot representing 1 to 2 Xist transcripts, or 1 to 2 Xist molecules. All right. So then with only 100 to 200 Xist particles on the inactive X chromosome, if we were to place the Xist particles end on end, there would only be enough Xist to cover about 1% of this 160 megabase chromosome. Or put differently, there's only 1 Xist molecule for every 10 to 20 genes. So that raises this question, because Xist is at a stoichiometric disadvantage, how does it actually silence an entire chromosome? And with respect to that, you see that there's sort of a discrepancy between what we're seeing at the single cell level by super resolution imaging and what we're seeing by chart sequencing, which really is measuring a population average across millions of cells. And so between what we're seeing in a single snapshot in time by chart and what we're seeing at the single cell level, we can deduce that Xist is actively moving around, very dynamically moving around the X chromosome. So that relates to its question of stoichiometry. So it turns out that Xist overcomes this unfavorable stoichiometry by recruiting catalytic factors. And these are factors that can amplify the work of Xist. So for example, here I'm showing 4 different catalytic factors that Xist is directly interacting with, including chromosome architectural factors, the cohesins, the SWI/SNP factors, Polycomb Repressive Complexes, as well as this non-canonical SMC protein called SMCHD1. And you'll note from this list of interacting proteins that Xist is both coming in contact with activators as well as repressors. And that is because, in fact, during the process of spreading, it is interacting with both the activating factors as well as the repressing factors. So we now turn our attention to the first function of Xist, which is the recruitment of repressive factors. And one of the factors that it recruits is this PRC2, or Polycomb Repressive Complex 2. This is an epigenetic complex that trimethylates histone H3 at lysine 27, and is a very important enzyme that represses gene expression and is important throughout development as well as during the etiology of disease. However, there has been a long-standing question in the field, not just about polycomb complexes, but about many, many other epigenetic complexes, about how they can be targeted to specific locations in our genome when these complexes are largely devoid of a sequence-specific DNA binding subunit. Okay, so how do they know where to go? Now, the answer to this question is going to be multifaceted, of course. There are going to be many different recruiting mechanisms, including transcription factors, including specific motifs in DNA and whatnot. But we believe that a major piece of the puzzle lies in non-coding RNA and sometimes even coding RNA, dating back to an experiment that we did 10 years ago in which we demonstrated that PRC2 can directly interact with RNA, in this case Xist, and recruit PRC2 to the X chromosome. And it's doing so through a motif at the very 5' end of Xist called repeat A. So this is a biochemical analysis that shows that PRC2 interacts with Xist RNA with high affinity, with a dissociation constant of 20 to 80 nanomolar, which is a very good dissociation constant for an RNA binding protein, and it contrasts with the affinities, or very low affinities, for these nonspecific RNAs from various other species like Tetrahymena and bacteria. Okay, so now in this slide, I will attempt to convey the complex dynamics that occur between the RNA and PRC2. So as we envision it, now initially, the RNA that contains this repeat A motif will attract polycomb repressive complexes to the X inactivation center. Okay. Now, very importantly, our genetic and biochemical experiments show that even though the long non-coding RNA is recruiting PRC2 in a site-specific manner, that does not mean that it is automatically going to load that complex onto chromatin, or that it will induce the catalysis on H3K27. Okay. So in fact, these steps are biochemically and genetically separable. So for example, as long as the antisense repressor, Cy-X, is expressed from the inactivation center, we see that this complex does not load onto chromatin. So at this time, we can see a RIP, RNA immunoprecipitation, between PRC2 and repeat A RNA, but we do not see them chipping onto the 5' end of Xist. And it is only when the antisense RNA disappears do we see that complex load onto the X chromosome. But even so, that does not by itself unleash the methyltransferase activity of the catalytic subunit EZH2. However, when this complex comes into contact with this accessory subunit called JARET2, we see that the affinity of the RNA for the catalytic subunit EZH2 decreases. So the affinity goes down, and that loss of binding of the RNA to EZH2 is associated with unleashing of the histone methyltransferase activity. So again, this principle illustrates how we can separate polycomb recruitment from its loading onto chromatin to its catalysis on H3K27. Okay, so take-home message, long non-coding RNAs can recruit PRC2, but hold their activity or hold the activity of PRC2 in check until a developmental signal is received, at which time PRC2 and JARET2 interact to trimethylate histone H3 at lysine 27. Xist overcomes its unfavorable stoichiometry by recruiting these catalytic factors. And we envision that these factors use a hit and run mechanism to silence the entire chromosome in an efficient manner. So again, Xist first binds to the inactivation center, nucleates at that site, recruits all these factors, and then spreads in 3 dimension to about 100 sites located across the chromosome. And then at these secondary sites, we're taking as an example here PRC2, but there are many other catalytic factors. So PRC2 lands, it processively methylates successive nucleosomes until it covers about 1 or 2 megabases of chromatin. And imagine that this happens 100 times over across the X chromosome, more or less at the same time. And then you can see how this process of silencing can be amplified and can take place in a very efficient manner. So now I'd like to turn your attention to the second aspect of Xist function, and that is its antagonization or its repulsion of the activating factors. And we're going to use as an example this epigenetic factor called SWI/SNF. Okay, so SWI/SNF is an ATP-dependent chromatin remodeling enzyme. And central to its activity is the catalytic subunit BRG1. So SWI/SNF is normally associated with open chromatin. Indeed, it makes chromatin accessible, poising it for gene activation. And so normally you would find SWI/SNF on the active X chromosome, but not on the inactive X chromosome. As Xist RNA spreads over the inactive X chromosome, the RNA comes into contact with BRG1 and inhibits the ATPase activity of BRG1, just as it inhibits the methyltransferase activity of PRC2. So this is an immunofluorescence experiment, and you can see that BRG1 is normally present throughout the nucleus. But where there is Xist RNA, shown here in red, you can see that there is a depletion of BRG1 over that same chromosome territory, suggesting that Xist RNA evicts BRG1 from the inactive X chromosome. All right, so then we'll now turn to the third and final function of Xist, and that is its role in directing changes in 3-dimensional chromosome architecture. So our RNA proteomic analysis also showed that Xist is interacting with a number of chromosome architectural factors. So you can see here various cohesins as well as CTCF. These are 2 architectural factors that go to construct 3D chromatin. So we've already talked extensively about CTCF, a zinc finger protein that brings together distant genetic elements that form these chromatin loops. And then we have cohesins, which are this multi-subunit complex that forms a ring around the base of the loops to lock in that architectural structure. So it is known that mammalian chromosomes are organized into 2 distinct entities, one called the topologically associating domain, or the TAD, and the other, the compartment. So TADs are these large loops of chromatin of around 1 to 2 megabases in size, within which genes can be, though they don't have to be, coordinately regulated. And then the active TADs, or that active loops, coalesce and form a separate compartment called the A compartment, whereas the inactive or the less active genes form another type of compartment that's called a B compartment. So as you might imagine, the inactive X chromosome is organized completely differently from all other chromosomes. So now here's an inactive X chromosome. The entire chromosome is shown across the top. And what I've done here is magnify the 2 ends of the chromosome. And you can see from this contact heat map that these TADs, which are these triangular structures, can be seen essentially all across the chromosome. So the active X chromosome looks a lot like any other chromosome, like all autosomes. On the X chromosome, there are about 110 of these topologically associating domains. Now contrast that with the inactive X chromosome, where, yes, you might still be able to envision a formation of these topologically associating domains, but they are much, much weakened. And so one of the things that Xist has to do as it's spreading across the chromosome is to attenuate the formation, although not abolish, attenuate the formation of these so-called TADs. When we mutate repeat B, so that's the critical domain for Xist spreading, we see that nothing happens to the active X chromosome. The active X chromosome still has 110 or so TADs. But on the other hand, when we do the same thing to the inactive X chromosome, you start to see that these TADs persist on that chromosome, or that maybe they're even coming back. They either persist or come back, suggesting that Xist is very important to the attenuation of these topologically associating domains. So how does Xist do this? Well, it's probably doing a number of different things, one of which is that it is also evicting cohesins from that chromosome. So this is one of the subunits of cohesin. It's an immunofluorescence that shows that cohesins are normally widely distributed throughout the nucleus. But again, where there is Xist, shown here in red, you see a depletion of cohesins next to this arrow. And the same is true of CTCF. Okay. Now, when we delete Xist, the cohesins come back, as shown here by this a red peak of cohesins here, and you can see that in the wild-type chromosome, those two red peaks are not present. Xist is not only attenuating TADs, but it's also directing the formation of these inactive X-specific compartments. So SMCHD1 is very important in the formation of these XI-specific compartments. And it was Emma Whitelaw who observed many years ago that embryos lacking SMCHD1 would die in mid-gestation due to dysfunctional X inactivation. So SMCHD1 is a non-canonical SMC protein that's like the cohesins and condensins, except that it has a very different function. So here you can see that Xist plays a very active role in the recruitment and the enrichment of SMCHD1 along the inactive X chromosome. And is in fact one of the proteins that we identified when we performed the Xist proteomic analysis as a factor that directly interacts with Xist. So it turns out that SMCHD1 plays at least two important roles during X inactivation. So in the first, it's aiding the local spreading of Xist PRC2 complexes. And in the second, it's merging these inactive X specific compartments. Okay, so here's the first role. SMCHD1 is important for the regional spreading of Xist, and you can see in this epigenomic analysis in the first track that Xist spreads along the, this region of the X chromosome, is about 1 megabase, more or less evenly. But in the SMCHD1 knockout, which is shown in the second track, you can see that there is a depletion of Xist across this 1 megabase domain, which is also green shaded. And associated with that is a defect in polycomb spreading and H3K27 methylation across that same domain. So SMCHD1 is very important for regional spreading of Xist, and the loss of SMCHD1 results in a defect of spreading as well as a focal loss of H3K27 methylation. Now, the other thing that SMCHD1 does is that it merges these inactive X-specific compartments. So shown here are high C experiments, and what I'm showing is a contact heat map for the active X chromosome in the wild-type state, with the active X chromosome shown here along the diagonal. And what you probably cannot see at this magnification is that there are about 110 topologically associated domains on that active chromosome, whereas on the inactive chromosome, those TADs are significantly weakened, and instead of TADs, this X chromosome shows two large so-called megadomains. So here's one megadomain, and here's another megadomain. Now contrast that with what happens when we remove SMCHD1, and on the active X chromosome, nothing happens. But on the inactive X chromosome, you see that these two megadomains start to break up. And that can be much better visualized by going to the bottom set of panels. These are heat maps of Pearson correlation coefficients. And what you can see is that in the wild-type inactive X chromosome, these two megadomains really pop out, so shown in red. But when SMCHD1 is removed, those two megadomains adopt a checkerboard pattern. Okay. And the checkerboard pattern that you see on the mutant inactive X is distinctly different from what you see on the active X chromosome, which shows a much finer checkerboarding pattern that's consistent with the AB compartments that we talked about a few slides ago. So we can better visualize this by going to a principal component analysis. And in the first principal component, you can see these red-blue structures on the active X chromosome, which are the AB compartments. And when we remove SMCHD1, nothing happens to the active X chromosome. It's impervious to there being a loss of SMCHD1. But then here's the inactive X chromosome, and you can see that in the wild-type state, there are these two megadomains, one in blue and one in red. But when we remove SMCHD1, those two megadomains break up into these finer structures that we call S1 and S2 compartments, referring to the fact that they appear only when we remove SMCHD1. But also appreciate that these finer structures are not the AB compartments that you see on the active X chromosome. So it turns out that these S1, S2 compartments are intermediate structures during the formation of the inactive X. So prior to X inactivation, we see these AB compartments like we see on all chromosomes. But then at the onset of X inactivation, as Xist spreads over the X chromosome, it merges, it being Xist, merges the red and blue compartments to form these S1, S2 structures, these larger S1, S2 structures. And as X inactivation proceeds, Xist recruits SMCHD1, and SMCHD1 in turn merges the S1, S2 compartments into these two megadomains to form a compartmentless chromosome. All right, so in these 3 lectures, I've thrown a lot of information at you, and what I'm going to attempt to do in this final slide is to integrate some of that information. So we envision that at the onset of X inactivation, this RNA with the repeat A motif recruits PRC2 to the X inactivation center. But as I mentioned, the recruitment process, the RNA is very important for the recruitment process, but it also holds the activity of PRC2 in check. So recruitment is not the same thing as loading, which is not the same thing as catalysis. Because as long as the antisense RNA is expressed, PRC2 is prevented from loading onto chromatin. And I also mentioned in lecture 2 that at this time, one of the very first things that we see during cell differentiation is a pairing of the 2 X chromosomes. And as a result of this pairing process, there's a mutually exclusive determination of the active and the inactive X chromosome, presumably through an asymmetric expression pattern of the antisense RNA psi-X. So from the future inactive X chromosome, the antisense RNA disappears, while the antisense RNA persists on the future active X chromosome. All right, so then on the future inactive X chromosome, the disappearance of the antisense RNA allows PRC2 to load onto chromatin, but again, that is not enough to unleash the methyltransferase activity of EZH2. When the RNA comes into contact with the accessory subunit JARED2, the affinity of the RNA for PRC2 decreases. The RNA is at least partially dislodged, and that unleashes the methyltransferase activity of EZH2. Okay, so while all of this is happening, at the same time, we see that JPX RNA, this RNA which is just upstream of Xist, is transcriptionally upregulated tenfold. And when it crosses a certain threshold, as it will do only in the female cell, because the female has 2 copies of JPX, JPX evicts CTCF from the 5' end of Xist. And probably around the same time, the Gribnow lab has shown that this transcriptional repressor, REX1, is degraded by an E3 ubiquitin lysase called RNF12. And it's really the combination of all of these events, in particular, the downregulation of the antisense RNA, the upregulation of JPX RNA, and the eviction of CTCF and REX1, that allows full-length Xist to be expressed for the first time. So Xist, of course, also has a binding site for PRC2, and we now know from RNA proteomic analysis that Xist probably binds to about 100 other proteins. Now, this RNA protein complex has to first attach or load onto a single nucleation site through YY1, the transcription factor YY1. Now, without attaching to YY1, this RNA protein complex will diffuse through the rest of the nucleus. So from this single nucleation center, the RNA protein complex then spreads in 3 dimensions across the rest of the X chromosome. And again, it does 3 things. It's not only recruiting silencing factors to the rest of the X chromosome, but it's also evicting activating factors like cohesins and BRG1, or the splice-NIP factor, from that X chromosome. So that, in a nutshell, is how we're viewing the initiation and spreading of X inactivation. And I should say, by way of conclusion, that this is a model. So it is a facsimile of what we can't actually directly visualize in the natural world. And scientists use these models as basic frameworks within which we can design additional experiments to probe, to refute, or to accept a hypothesis. And of course, scientists disagree all the time with each other about exactly how things are working in nature. And so we hope that additional data that we will generate in the coming years will allow us to continually refine this model. And I hope that I'll be able to share some of those new ideas with you in the coming years.